Perfect. Yeah, thank you all for joining today. The Olink team has already introduced themselves, so we'll just pass it over to them to take it away with their awesome technology on proteomics. And if anybody has any questions, please put it in the chat or in the Q&A, and we'll either answer it as we go, or we can answer everything at the end. So take it away.
All right, thank you, Alex, and I apologize for all the issues, but thank you so much for allowing us to take part in your webinar series. My name is David Getz. I'm a BDM at Olink covering New York, New Jersey, Pennsylvania region. The goal in this presentation is to share with you who we are as a company, present our service and capabilities, and share some pertinent case studies from our scientific affairs team. So please, while we're going through our presentation, feel free to type any questions in the chat so we can be sure to answer them during the Q&A. Next slide, please. So at Olink, we have emerged as a strong market leader with our technology, helping to overcome limitations we have seen in the world of proteomics. In just a few short years, Olink has grown rapidly from a small Swedish-based biotech company offering assays for a few hundred proteins to a NASDAQ-listed organization with a strong global presence. We have a broad portfolio of flexible protein biomarker solutions and a library of high-quality, thoroughly validated assays. We're a company that offers analytical services in our Boston and Uppsala locations, and Uppsala being our headquarters. You can send your samples into either locations and we'll deliver the data to you. And we also are a kit manufacturer and an instrument provider, so you have the option to run samples in your own lab or with an external partner or CRO. And looking down at the 8 boxes on the bottom, we're very proud to serve all the most prestigious research organizations within academia around the world, as well as the top 20 biopharma companies. We have demonstrated strong scientific validations with more than 1,100, close to 1,200 now, peer-reviewed publications and new papers coming out every day. Our technology is scalable. You can advance your findings into clinical decision-making. As an example, we've worked with the UK Biobank. We ran about 58,000 samples across 1,500 proteins in about 20 weeks using 3 NovaSeq, so that was pretty incredible. And next slide, please. So just to go through some of the limitations we have seen in the past that led us to develop our superior PA technology, we've seen poor sensitivity, lack of specificity, high sample consumption, and limited dynamic range, which of course has led us to develop the PEA technology. And yes, thank you. So now let me tell you how the Olink solution can help mitigate these limitations we've just discussed and we've seen. Our simple but elegant PEA technology has helped bridge the challenge of proteomics. We have solved the challenges of protein multiplexing by able to achieve exceptional specificity, high sensitivity, precision, and a large dynamic range and throughput. No other technology or platform solves all of these challenges simultaneously, which is why the market has readily adopted PEA. And next slide, please. I know that most of you are users of mass spec, and I thought it would be a good opportunity to present with you some correlation data to show how mass spec and our PEA technology can work hand in hand. So displayed on the right, on the blocks on the right side, are some correlations that were performed both by King's College London and Helmholtz. Both perfectly illustrate the complementarity between 2 platforms, showing that mass spec is a great methodology, but skews its detection towards the most abundant proteins, whereas Olink's highly sensitive targeted panels can measure many analytes missed by mass spec. Next slide, please. All right, so let's jump into the core of Olink's technology, the PEA, Proximity Extension Assay. It's what has enabled us to be so innovative in the biomarker space. So PEA employs dual antibody recognition, which drives incredible specificity in our assays. Coupling this with our PCR amplification, it enables robust sensitivity. The PCR amplification of the hybridized antibody pairs on the protein that you see transforms detection of antigens into unique DNA barcodes that you see right there. This allows for extremely high assay multiplexing in very small volumes. We use minute volumes to run our 4896, 384, and 3072 plex flexibility. And of course, all of these are scalable from discovery to clinical implementation. Thank you. So using PEA technology, we were able to develop a robust portfolio with NGS and qPCR readouts. So on the left side, we have our NGS readouts of Explore 3072 and Explore 384. And the Explore 3072 is built up of 8 different 384 panels. And then moving to the center, we have our more targeted approach of Target 96 and Target 48. And on the right side is our Olink Focus and Olink Flex products. Focus allows you to custom develop a panel and to use in your lab for up to 21 proteins, whereas the Flex is a custom mix and match panel of 14 to 21 plex, where you can choose from 200 proteins that we have pre-validated, which is setting a new standard in protein analysis. And now all the 4 qPCR assays here can be run on the Signature Q100, which is an amazing benchtop instrument that was specifically built for the PEA. On our platform, we have the ability to be scalable up and down in plex, so your studies can begin with a broad discovery outlook of 3,000 proteins. That leads to a more narrow targeted approach of 96 proteins using a target assay. From there, you have the ability to really focus your research question to a customized view using our Flex products and Focus products. So I wanted to go through a quick workflow just so everyone can see how the Target and Flex and Focus panels work. Initially, there's an immuno reaction, which is 16 to 22 hours overnight. Then you have your extension pre-amplification for your PCR step, which is 2 hours. And then for this, since it's run on qPCR, you would put this on the Q100 instrument, and that runs about 3 hours. So you get results in 24 hours. I don't have the slide to display for Explore, but I'll quickly go through. The difference with Explore, you would run your immunoreaction, your extension, and then you would get, your next step would be your library prep, which would take about 4 to 5 hours. And from there, you would do your NGS readout, which is about 10 hours. So with Explore, you would get results in roughly 36 hours with 5 hours of hands-on time. But it's pretty incredible because in 1 week on one NovaSeq, you can get about or more than 1 million data points per instrument. Next slide, please. So an added strength of our PEA technology is the flexibility of input samples, far beyond EDTA, plasma, or serum. We've worked with 1 microliter per 96 and 384 panel, and you use 6 microliters per 3072 panel. And on the left side, you see all the matrices that we've worked with. And of course, we have a mouse exploratory platform and panel that you can utilize on Target 96 using only 1 microliter. Next slide, please. So now that we have discussed our platforms and our assay portfolio, I want to switch gears and move on to our pre and post analytical data tools that we have to offer. Olink Insight is our new and fantastic digital online tool that is useful for a variety of concepts. Whether you want to use it for pre-study design, you can ensure you're properly planning your study before you even run a sample. It can be utilized for pathway browsing, sharing protein lists, finding the best matches to Olink panels that we have. You can view normal protein ranges and disease atlases on there as well. Unfortunately, we can't dive too deep into the details of what the platform can offer, but we would be more than happy to set up a call at a later date to demo it if anyone has interest in that. You're also welcome to click on the website, or not click, but go to the website insight.olink.com, and you can click around and check it out there. All right, so beyond our analysis services, we're able to assist with a broad array of Olink support. We support our customers all the way from study design and sample analysis to actionable insights. We first start by providing normalized proteomics data and an analysis report if you choose to run in our analysis lab, but if you run it in your own lab, you'll get the data as well. In addition, you have free access to our Olink statistical analysis tools using the Shiny app for data visualization. The app provides a point-and-click interface where the user can perform t-tests, ANOVAs, PCAs, and generate volcano plots and heat maps. The app operates in your web browser and can handle all of the data, or all of our data. It is built on a restricted set of functions from the Olink Analyze R package, so you can download these on GitHub and CRAN. We also have an in-house biostats and data science team that can assist with any questions along the way that you have if needed. With all this support, we can help guide you to publications, clinical trials, and grant submissions. So scalability and quality are two key features of our Olink PEA technology. With our flexible product portfolio, you can now run midplex, 40 samples, to high-plex with 384 samples for 3,000 proteins per kit. So whether you need a protein signature or specific insight into a biomarker, we encourage you to take us on board on your multi-omics journey. You can rely on Olink as a trusted, flexible, and transparent partner. We are a great company that's easy to work with. We provide great high-quality data, and we offer white glove service throughout your whole journey of proteomics. So we hope that you want to learn more, and after this call, maybe set up a couple more meetings to dive deeper into our products. Now we'll have our scientific affairs team present some case studies and move on to the Q&A after that. Thank you.
Thank you, David. Hopefully you can all hear me. Wonderful. So I'm Mariana Fonch, already introduced myself, part of the scientific affairs team looking after neurology. And today I'm really excited to share with you a few examples of how the technology is being used to address important scientific questions in the field of neurology. On the next slide, please, we have just here a representation, as David mentioned, that with our technology, we can cover really all the major biological pathways. And this is important for neuroscience in particular, because there is a complexity and a multitude of aspects that are involved in complex neurological disorders. And I'd like to highlight to you now on the next slide, please, just a few molecules which you might recognize if you're working in the neuroscience space, where we have a broad coverage of cytokines and inflammatory markers, but also synaptic and neuronal health markers that can help you with your research. And importantly, in the next slide, I can show you that not only we have a broad spectrum of assays covering very important proteins, but actually we are not compromising on the sensitivity and the specificity of each assay. So here is an image from a published study by Alnylam Pharmaceuticals, where they were able to identify that neurofilament light is a strong biomarker to look at a rare neuromuscular disorder called HATTR. Now, neurofilament light, as you might know, is a very well-established and known neurodegeneration marker. And here they compare the performance of this marker in the Olink assay versus the gold standard, traditional gold standard Simoa singleplex. And what you can see is a beautiful correlation between the results with a correlation of 0.96, really showing that the Olink assay has a very strong performance and very high sensitivity. Now, the advantage of using Olink, if you're interested in getting an understanding of more and a broader pathway coverage, is that with a very small sample size, sample volume, you can also get an additional insights for more than 1,000 proteins. So on the next slide, I want to show you here, just represented by a few examples of how it's really not surprising, given the breadth of the assays as well as the quality, how there's many publications using Olink, leveraging Olink to advance neuroscience research. And here selected just a few, providing examples across different therapeutic areas, such as ALS, MS, Alzheimer's disease, but also using different matrices, such as plasma, CSF, extracellular vesicles, looking at mouse or human matrices, and really showcasing that whatever your field of research within neuroscience is, we probably already have some references that you can use to move forward your research. And today, very shortly, thank you, very shortly, I'd like to just give you a few snapshots about 3 case studies that address the question of how can we improve disease classification, treatment response understanding, and also disease biology understanding using Olink. This is just to pique your interest. These are published studies, and you see the references here, so you can, so we can share them with you, and you can look in more detail. So in this first example, this is from a study driven by the labs of Professor Charlotte Tennyson and led by Martha Del Campo, which was recently published in Nature Aging, and they really did a large screening of the CSF proteome, and they were able to uncover very strong panels for diagnostic and differential diagnostic purposes. So they started by doing this large discovery effort, looking at more than 1,000 proteins in the CSF of patients across the Alzheimer's disease spectrum, and also looking at patients with dementias that were not Alzheimer's disease, so non-AD dementias. And they were able to identify several proteins that were altered in these conditions. And through classification modeling, they were able to really narrow it down to 2 panels that have very strong performance in terms of being able to differentiate Alzheimer's disease patients from healthy individuals, and also Alzheimer's disease versus non-Alzheimer's disease dementia. And that's what you can see on the right. You have a representation of the area under the curve, which is just a measure of the quality of a model's prediction, in this case for disease classification. And you can see they get very strong classification ability of 0.96 for Alzheimer's versus control, and of 0.87 for Alzheimer's versus non-Alzheimer's disease dementia. And here we would expect a slightly lower classification ability, as there are a lot of overlapping processes across the different types of dementia. So just a beautiful example also of going from discovery to clinical translation, as David mentioned previously. And also using different product ranges, so starting with several target panels and then narrowing down to focus applications. So on the next case study, I would like to just very briefly highlight to you an example when Olink Proteomics was used to investigate and to find strong biomarkers for a rare neurological disorder called Niemann-Pick disease type C1. And this research was very important, as they're still lacking FDA-approved treatments for NPC1. And the authors were really trying to identify good markers that could be used in clinical trials to assess the response to treatment, and also to be able to monitor these patients over time. And as you can see on the right panel, they were able to identify not only established proteins and validate established proteins in this disease, but also to identify several new proteins altered in the CSF of these patients. And what I show you on the small graphic on the right is their validation, in this case, using ELISA. So you can see that our technology is also very well translated into other orthogonal methods. And finally, on the next panel, thank you. Very importantly, they were able to correlate these markers with important clinical phenotypes. So you can see in this case for one of their biomarkers, CCL18, that it correlates strongly with age of disease onset and also with the progression rate of this disease. So they highlight that this information can really be strong in moving forward to better clinical trial designs so that we can get a treatment FDA approved for patients with this rare disease. And finally, on the last case study before I pass to Michael, I would like to highlight to you an example in the space of Parkinson's disease where researchers were using the Olink technology to gain further disease biology understanding and identifying potential new Parkinson's disease therapeutic targets. So here, instead of going into a large discovery effort, the authors were really trying to narrow it down to processes that are already known to be involved in Parkinson's disease, such as inflammation and cardiovascular processes. And for that, they used 3 panels, Olink panels of 96, cardiovascular, inflammation, and the neuroexploratory panels. And they did a deep dive into the profiling of patients with Parkinson's disease. They were followed over 6 years into their disease progression journey. And they were able to, with this approach, to elucidate several new disease mechanisms, including being able to understand that there is an increased thromboembolic risk and neuroinflammation that they expect to be involved in the increased aggregation of alpha-synuclein. And very importantly, I think also relevant for some of you here, there are several open sources that can be used, and the authors in this study compared their results and were able to validate their results in an existing data set on Explore 1500 from the PPMI initiative. So just a great example that having resources out there and more than 1,000 publications with Olink can be a great resource for your further research. And with that, I pass it to Michael and be happy to discuss afterwards. Thank you.
Yeah, thank you, Mariana. So, what I will do is, next slide, please. I will try to introduce a case that is more broadly applicable. So it's for a rare lung disease that has rather devastating consequences, and then show you what people using Olink have done to actually derive a clinically relevant biomarker signature. So the work has been published in Lancet Respiratory Medicine, was undertaken in Justin Oldham's lab at UC Davis, and the first author is Willis Bowman. Next slide, please. And what they were interested in is in this rare kind of like lung complication called progressive fibrosing interstitial lung disease. So it's rare because the incidence is just 13 to 20 per 100,000. And it is a complex syndrome rather than a single disease. So anybody who's speaking to a pathologist when they don't know what to call something exactly, they call it a syndrome. It is a comorbidity for various connective tissue pathologies. So rheumatoid arthritis and systemic sclerosis, and it has rather ubiquitous symptoms. So it's relatively benign and hard to diagnose, to actually achieve the diagnosis of IPF. What will give it away is shown in the top left corner is this clubbing of the fingers, and what will happen pathophysiologically is that there is a progressive scarring of lung tissue that will basically impede the gas exchange in the lung. So the associated complications are twofold. If you have this kind of disease, first you will get problems with the right side of the heart. So you will develop pulmonary hypertension, right-sided heart failure, so-called pulmonale, and you will also have a massively increased risk of lung cancer and respiratory failure. And you'll survive up to 5 years after initial diagnosis. So it's hard to diagnose, it's hard to give good prognosis, and it's really, really bad. So the key question that Willis Bowman asked is, next slide, please. Can we find biomarkers for disease progression? So how did they go about this? The study design was as follows. They had 2 different cohorts, a discovery cohort, they had a validation cohort. One collected in California, one collected independently in Texas. They had all different forms of ILD covered. They had long-term medical outcome, and they had a hypothesis. They basically said, hey, inflammation is going to play a role, so let's use one of the Olink inflammation panels to address this question. And then they had a rather elaborate and fancy machine learning algorithm to do their bioinformatics analysis. So what did they find? The results were relatively straightforward. So in the discovery cohort, they found 31 biomarkers that associated with the progressive form of the disease, of which 17 were validated in the validation cohort. And then they did their lasso logistic regression, and they could basically identify a core protein signature of 12 proteins that's shown here, that basically accurately ascribes risk to developing the progressive form of interstitial lung disease. And this had a sensitivity of 0.9 and a negative predictive value of 0.91, which is great because that's actually a clinically relevant, a clinical grade biomarker signature, because only 10% of the patients with the risk signature or with the low risk signature will actually develop the progressive form of the disease. So next slide, please. And then in the discussion, they actually say that a trial with a 1-to-1 randomization would normally require almost 700 patients to detect a 50% reduction in lung function. However, if they would design a trial based on this 12 protein signature, that actually would reduce the patient number by 80% to roughly 140. Next slide, please. So intermediately synthesizing, the message is you can absolutely use us to develop biomarker signature even for rare types of diseases. It has implications for clinical screening, and if you're interested in running trials or you do develop biomarker signatures for clinical trials, it can reduce both the cost and the time. And the second part of my presentation, next slide, the question is can we valorize this study? So this is really for those researchers among you that collaborate a lot with pharma. Next slide, please. So here are the clinical trial costs for respiratory diseases, including ILD. So you see it's between $6 and $30 million that phase 1 to phase 3 trial on average costs. And with the assumption there's a reduction of the trial size by 80% will also lead to a reduction in price. Next slide, please. These are actually the potential cost savings that you could realize implementing this signature into the clinic. And then on the next slide, and this actually has real-world ramifications, because here is a real-world example of a clinical trial. That's the InBuild trial. It's a pivotal trial because it changed treatment of IPF in the clinic. It's double-blind, placebo-controlled. It's a medicine originally developed by Boehringer Ingelheim. However, for this trial, they had to read out, or they had to observe patients for over a year, and only those that basically were followed for a year could be included. It was a worldwide trial. They screened 1,000 individuals. They enrolled only roughly 60%, so 660. And of those, 20% discontinued nintedanib due to due to side effects. So you can actually say that there is a massive amount of patients that has been overtreated in this trial and never had any chance to basically benefit from the potential therapeutic effect. And with that, next slide, we actually come to the end of my presentation, because price is what you pay, but value is ultimately what patients get, paraphrasing Warren Buffett. And with that, I'm happy to take questions.
Perfect. Well, thanks for the great presentation. Yeah, if anybody has any questions, you could either raise their hands, put them in the chat or the Q&A. We can answer from there. I have one to kind of start off with. It's awesome seeing all the clinical relevance for everything here. But I know a lot of our researchers work with non-human organisms. Do you have panels available for like mouse, rat, any other like bacterial, for especially like the custom ones?
Yeah.
Yes, go ahead, Mike. Yes, we do have a mouse exploratory panel, which you can use, and we would be happy to share with you also some publications where there has been studies. So it's a broad panel that tries to cover a lot of the diversity of biological processes, so that it can be so that you can use it for your research.
Okay, cool. And what about like other custom organisms that people may be working with? Is that available yet, or is that something maybe in the roadmap a couple years down the road?
So we have done the comparison, for example, for non-human primates, and we do know that for the majority of the assays that we have, in terms of the panels we have, there is a very strong correlation with the human plasma performance. So we know that for that matrix is definitely possible. And for others, what we try to do is some homology analysis and see if it's suitable. And of course, we try to work with researchers that have experience with the model system to see if the particular area of interest could be suitable. But we do not have targeted panels for, let's say, Drosophila or zebrafish, not yet at least.
Okay, cool, thanks.
We also have quite a lot of research done on iPSC derived cells and other in vitro systems. I don't know if that's something that within the institute there's interest.
Yeah, no, there's definitely some people who work with different in vitro systems, like you said, are iPSCs. There's actually a fair number of people who work with those types of cells.
Wonderful. So in that case, we can use, it has been used, for example, to look at the supernatants, so to look at secreted molecules, but also you can lyse the cells and run them. Really, the most important is that we do not, or I'll leave that maybe to Ray to comment on it, but it's difficult if the cells or the tissue is fixed. That's when it's more challenging for us to run the platform.
So, okay, there is a question from the audience. Let's let the audience ask the question first. Jason.
Yeah, well, his question is, can we please comment on post-translationally modified proteins?
Yeah, sure. So I mean, we don't have a particular focus on post-translational modifications as of yet. It's if you I mean, you've seen how the assay works. It's in theory it's possible to basically, as long as you have a good antibody that recognizes the post-translational modification, you can then have a second one and specifically recognize that. But this isn't the focus right now. What we have done is we have looked at proteins, so we have looked at components of heteromeres. So if you don't look at like the standard post-translational modifications like phosphorylation or something like that, or glycosylation. But if your protein forms heteromeres, especially in circulation, so we sometimes have antibodies that specifically look at that. So specific targets, I mean, especially cytokines, for example, or things like that. So that you can actually relatively easy use the library for. And then the other thing is we're also not specifically geared towards splice isoforms. So it's always because the initial validation step is in serum and in plasma for global targets, and in CSF for neurology targets. We kind of like tilt towards the splice isoforms or the proteoforms that are relevant or seen in those two biospecimens.
And I would add that that's actually one aspect where it's very complementary to mass spectrometry. So we do know that several researchers are using mass spec to look at post-translational modifications and to look at isoforms, and then complementing and really working with Olink to look at more the lower abundant and sort of broader range of non-modified proteins. So I think they're very complementary technologies that fit very well together.
So thank you, Mariana and Michael, for the great examples. I enjoyed your examples very much. I have three very simple questions. Number one, regarding your reagent validation. How do you validate your antibody? Make sure that you are detecting what you think you're detecting, but not other things. Number two, reproducibility. And you showed a lot of clinical examples. How reproducible are the data across different labs? Especially you're selling this reagent to different academic labs, and based on our previous conversation, there are a lot of steps. So how reproducible are the, also, including both sample prep and then NGS labs? So how do you ensure that people from different labs get similar results with identical samples? And number three is a question for Michael in terms of a cost versus value. And I'm sure some of the attendees here are interested in learning that aspect of things. Thank you.
So for more on the technical side, perhaps I would like maybe Ray to share some of his insights on the aspect. I think he'll be better positioned. Otherwise, I'll answer, of course.
Well, I'd be happy to talk about a little bit about our validation process. So with every assay that we develop, we go through many different iterations of primer and antibody combinations before we pick the right one. And then we have an extensive validation process, which would take probably another half-hour meeting to go through everything we do. But I would be happy to chat and go through those at a later time in more detail. So, of course, the validation process, includes specificity, making sure that everything's working well, whether it's simplex or multiplex, going through all of those sort of steps, and it takes quite a bit of time. So it is a very involved process. So that was your first question. Could you repeat the second?
Second question is reproducibility. If the same clinical sample that Michael and Mariana presented in those papers are given to different labs across the world, do you have data supporting the reproducibility of the whole workflow essentially?
Sure. So when we have correlation studies with partner labs that we've done. Every lab that gets set up to run goes through the same exact process, where we train on site. We have them run the same sample that we run at all of our sites, as well as at our initial site, and then we do a full CV analysis to make sure that the assays are performing exactly the same at every step of the process.
And I would also add that there are several studies, several publications that compare the results. And for example, in one of the cases I shared for Parkinson's disease, they were able to actually find the same results looking at different cohorts. So one was looking at TANOPA and the other one was looking at the BMI, and they actually found the same insights in different cohorts. But also similar, if you look, for example, the work of Nancy group in Alzheimer's disease, they also find similar results to the group of Biofinder in Sweden in a different background. As long as the matrix is the same, the results seem to be quite comparable. But of course, it's very important, the conditions, the cohort backgrounds. But matrix is, I would say, from at least my experience reading literature, is the most important factor to look at that correlation.
Yeah, and then adding on the validation side, like we have been used extensively in large cohort studies. So basically, the UK Biobank is one example. We have a proteogenomics consortium that's called Scallop. That's another one. Decode Genetics in Iceland uses us extensively. And they've all published data concerning like surrogate measures for specificity, basically cis-pQTLs. So they have large cohorts, they do GVAs, and they find that in the absolute majority of cases, so more than 80%, if they test these large cohorts, they actually find a hit that coincides with the genomic locus of the protein that they're fundamentally measuring. So you can be very, very sure that once you hit SNP that controls expression, that maps to the locus, you actually measured that protein. And so that's to like independent hypothesis-free validation. And then lastly, concerning price, that's actually a question I need to punt to David, because we usually scientific affairs, we don't do pricing. So anything concerning pricing, that's for the sales team.
Just before pricing, I've also just added a link to the chat where you can see the validation that some of the validation that we do internally, and you can search through that aspect on the website, and you have a lot of different examples on specificity, precision, and intra-assay variability as well.
I guess, can you repeat your question on pricing? Was it more of a pricing question or like a value of how much something would cost?
It's a question for the attendees. So some of those attendees use our mass spec-based proteomics services. So we have certain cost on those type of services. So I guess they are very interested in, if they want to do your technology, how much it will cost to buy the reagent, do it in their own lab, how much will cost them to send it to your service lab, and what kind of value they will get for those, for those advanced technologies.
Sure. So, Anders, I don't know, do you want to discuss pricing on the Q100 and the target kits, since they seem to be more directed toward mouse panels?
Absolutely. So the Signature Q100 is going to all in come in around $100,000. And then in terms of kits for Target 48, starting at around $4,000. For Target 96, it's going to be around $6,000. And I don't know if we touched on this. Target 48 is going to be 40 samples, and Target 96 is going to be 88 samples per kit. And in terms of Flex, that's also going to be, which is the one of the customizable panels that you can order. In terms of cost there, it's also going to depend on the number of the plex. So whether you're going with 15 to 21. But that's going to start at $4,100 per kit. So just to offer a little bit of clarification there on pricing. I don't know, Hong, if that helped a little bit there, but happy to have individual conversations as well.
Yeah, I'm just trying to see who my competitors are and how much they charge. That's all. But yeah, customers are very interested in that type of things. So.
Yeah, and for the value piece, if you look at the library a little bit in depth, especially at the Explore library, so this is all for human samples, we do have 60 high abundant proteins in there. So proteins that you would generally pick up in any like mass spec experiment, irrespectively of the technology you use, especially on serum or plasma in terms of initial validation. But then what people have looked at is like the types of additional, like what in addition you cover, and you actually cover 9 orders of magnitude more when you add the Explore library. So especially there are a lot of like low abundant proteins that you will actually have that you will have problems to pick up with mass spectrometry. So at the same time, combining the technologies and showing the complementarity, you add on the low abundant protein And at the same time, you will always have an automatic validation of the data you actually received with your mass spec experiment. So that's kind of the additional value.
And we showed you here, I think, throughout the presentation, some comparison with ELISA, Simoa, with mass spec. But if you would like also other publications that independently have compared us with other technologies, those are definitely out there, and we can share them with you for visibility. If you have an interest, would you be able to share which type of technologies you're also considering, or?
No, we are only doing mass spec, and we obviously met previously about Olink and also SomaLogic, and my personal preference is antibody other than aptamer for very good reasons. So it really depends on Alex bandwidth. So if Alex has the time to do bioinformatics, then we are happy to talk further. If he doesn't have any bandwidth, then we can't really bring it in.
Yeah, that's always the $1 million question. But that also, to answer for the Olink team as well. I mean, you guys do have a analysis platform that you can use, which is always good to kind of have as something to ease a little bit of the bandwidth and spread that weight out a little bit.
Correct. I mean, and we have the R packages that can be used, but also now on the new platform that David mentioned, the Olink Insight, a lot of the information is already there for references and for both the planning and the analysis. And then, of course, we also have it as a service, data science as a service. So that's another possibility if you're just getting started.
Any other questions before we wrap it up? No? Okay, cool. Well, thank you everyone for the great presentation. If anybody has any questions, of course, we could follow up with either myself, Hong, or anybody from the Olink team, and we can connect everyone who needs to get connected and keep going forward. So thank you very much, everybody.
Thank you all. It was a pleasure.
Yep. Have a great day.
Thank you.
Have a great day. Bye-bye.